bit louder. Hello, everyone. We should start to settle down. We do have to keep to some kind of a schedule. We found a set of car keys. Sarah is waving them in the air.
If someone's missing their car keys, come to me. I got them.
Okay. So I'd like to, it's about time, as we say, to introduce the second speaker, Michael Long from NYU, who has really studied time in the nervous system, and I think he's going to tell us quite a lot about it, using, in some sense, the same experimental system that Dan has introduced us to, but from another perspective.
Yes.
Okay. Take it away.
So I told Dan that my talk is just going to be everything he said, but with the word "not" in front of it. Just kidding. Dan is an incredible scientist, and I don't think I'd be here without his work. And it was an extremely thrilling talk. I don't think I've seen the story yet. And beautiful stuff. Thank you to David and Randy for being invited here. So my question today is, how can sloppy neural circuits keep precise time? And so I guess, as David stated it, this is one of the big questions for what we're trying to, I guess, address over the next few days. How's the world represented in the brain? And I think one challenge to having any kind of truthful representation of the world is neural noise. And I think, as we all know, the brain is a pretty noisy place. If we look at all these components, they're pretty unreliable. Everything from sensory transduction here at the level of photoreceptors, for example, electrical noise, stochastic opening and closing of individual ion channels, to networks, synaptic noise. The probability of release is not one. These things are probabilistic, as well as motor noise. So the whole thing seems quite unreliable. So how can we get anything done? How can we have any truthful representation of the outside world? This is a very simple experiment that I think many people know. Maybe some of the graduate students don't know it yet. This is kind of a classic paper from Zach Manin and Terry Sinofsky many years ago. And what Zach did is he patched onto one of these neurons in slices, think of a Campbell cell, something like this, and gave this complex waveform and found that across trials, spiking can be actually extremely precise. These cells like rapidly rising inputs. And so what we've learned here is that spike generation is not noisy. So it must be something else. And so this was a very intriguing paper from Mickey London and Michael Heuser a few years ago. And they made the case that networks themselves can be quite fresh. So even though individual cells can follow pretty faithfully their inputs, networks themselves, maybe because of things like probabilistic release and other factors, can be fragile. And this was a cool demonstration, I thought. They can add a single spike here and use a kind of clever approach to look at the network to ask, what's the impact of adding an extra spike into your network? And what they found is that just adding one spike causes this kind of avalanche that ultimately leads to 28 additional spikes being born into that network. And so this is a large amount of intrinsic noise, and that's then supportive of a rate curve, as opposed to thinking about spikes as anything other than something that we can add up in bulk. But that doesn't quite square with some experimental results that have been fairly consistent, although somewhat on the margins of the literature. And this is a really nice example from Christian Machins, working with Mike Weir and Tony Zador. And what Christian found is that when he records from rat auditory cortex, so this is back when rats were actually used in the lab, and there was a time, and they did whole cell patch clamp recordings in primary auditory cortex, and they played this complex stimulus a number of times. This is just the song of a Knudsen's frog. And when they did that, you can see across all of these trials this really beautiful, very precise, even at the synaptic level. Right? And A1 is 5 synapses in from the periphery. And yet, this information is transmitted quite truthfully at the level of this individual neuron. So not everything is really adding up. And I think one thing that's complex about sensory systems, and I'm certainly no expert, but of course, the idea of active sensation. We don't passively take in the world, but we're actively grabbing information from the outside world. And so if we can control for that, and this was, I think, a very clever experiment from Yves Freignac's group, where they controlled for it simply by, in this clever way. So they looked at the eye movements of a cat, and you can see these kind of chronic eye movements all around this complex scene, which happens to be another cat. Then they paralyzed the eye of this animal and moved the world across the eye to try and simulate the cat looking in exactly the same place 10 times in a row or so, all while patching a single neuron in primary visual cortex. And when they did that, I hope you can appreciate that this is just taken out of one part of their data. I hope you can appreciate that this is really a staggering amount of stereotypy across trials. So just at the level of all of these little synaptic events, this cell is really experiencing the same thing. So I thought the brain was a noisy place. Why is it that we now have several synapses in something that really seems to be not at all noisy, quite deterministic? And this is one of my favorite examples. This is from Kurzfeld and others up at the University of Washington. This is back when they could record single neurons with glass electrodes in human patients. This is tissue that would be later resected in the temporal lobe. So this is 6 or 7 synapses in the auditory system. This is a single neuron's responses to complex words. And the experiment was very simple. The patient was there. They were recorded from this neuron in superior temporal gyrus, and looked at the responses to listening to the word telephone twice, the word hamburger twice, corkscrew, etc. And what was really striking to me is that when this person heard telephone, hamburger, airplane, rattlesnake, the cell was totally silent, no spikes. When this person heard caterpillar, there were spikes, and these spikes look like, this is an extra spike here, but this spike looks to be around the same time. And then when they heard corkscrew, heard it twice, you can see this really beautiful precision that exists in an awake human in this very complex place across trial. So this cell is very selective. It doesn't fire here, and it fires with alarming precision there. And in the songbird, we see the same thing. So this goes a little bit to what Dan was talking about. This is a neuron that drives the song. And this is not one that projects to basal ganglia, but rather to the descending vocal production pathway, so-called RA projecting HVC neuron. I recorded this cell while the bird was singing this 3 times. So we put a small head-mounted drive. We could lower down electrodes and record the membrane potential of the cell while the bird is hopping around his cage and singing to his female. And what's interesting, you can see from these 3 trials from the same cell, that firing happens at one moment in this kind of high-frequency burst. But we also have a lot of synaptic activity, and I've blown it up for you. Just blown this up by an order of magnitude and laid these 3 trials right on top of themselves. And you can see this, almost quantum precision that exists across trials as the bird is producing his song. So if the brain's a noisy place, how can we get to something like this that looks so precise across trials? Dan did an amazing job of introducing the songbird, not only to me as a scientist, but to you as an audience. This is a movie that I hope works, of a male singing to a female. The male's got his head off screen, the female's here. You can get a feel for how this looks. And so we can take these pieces out, and the behavior itself is really a finely tuned thing. I think that's a point that Dan also really brought home as well. That these 3 epics here, here, and here really look amazingly precise. So we have precise circuitry that allows for this kind of precise behavior to occur. I think Dan did a reasonable job of explaining this as well. So there's a forebrain with two main kind of motor cortical-like regions. There's this HVC area. I'll be talking about that today. RA, which is kind of a primary vocal motor cortex. UVA is thalamus, these other areas. I've just put these bubbles on here because all of them, if you knock them out or you inactivate them, the bird's unable to sing. So these are all necessary components for a song to occur. And there's one central place, this HVC region, which is where song-related neural sequences are primarily formed. And I can go through the data that support that statement if anyone's curious. For time, I had to take those slides out of this talk. But one thing that's really clear, this is a kind of a more ensemble view of what I've shown you before with the intracellular recording. And these are data that Dan also showed in a different way, that this network actually forms a sequence of activity. So what you're looking at here are 8 premotor neurons. Each neuron is shown in a different color. This is an adult who's perfected his song so he can sing this thing pretty flawlessly. He's done 100,000 practice trials to get this just right. Such that this green cell, across all 10 trials, makes a burst, and the center of that burst jitters by less than a millisecond. So that's a staggering amount of temporal precision. And just to give you a sense of scale, there are about 40,000 of these cells. So there are 8 represented here, but a very large number of them that actually generate the song, and we think that there are about 200 neurons active at every moment in the song, and we'll come back to that. So the appearance of these kinds of network sequences appears to be the rule rather than the exception. And I think as people start to look using large channel count electrodes in behaving animals, they see this more and more. These are data from Yuri Buzaki's lab, from Chris Harvey's lab at Harvard, Takaki Komiyama's lab, Mike Palasa, a number of other people, see that the principal activity of these neurons forms what appears to be in a network these kind of larger scale sequences, such that the activity of each neuron is shown in one row, and as a collective, they form something like this. So the question is, how are these precisely timed sequences represented in the brain? So I thought that the brain could kind of cheat a bit, be able to maintain its millisecond precision simply by building into this some kind of spatial structure that could give it a leg up on making these millisecond precise bursts possible. And so one idea when we started thinking about networks, these are just all little neurons here, and I've schematized this and just describing one possibility by coloring this group blue, this group green, which happens at a second moment in time, and this group red. And so one idea is that there's a chronotopic map, that there's columns of cells, and that's one easy way of getting this kind of rigid temporal structure that's seen during the behavior. There are a number of other network organizations. I think on the walk here we talked about how difficult it is to describe what large networks are doing. Maybe we can learn something about that later, the right kind of language to discuss these. You can imagine another case where there's a wave-like propagation, maybe a group of cells here that then travels in some kind of propagating way across our cortical space, or some kind of mixed complex representation, or one that we just simply can't describe. So what we did to get to this is we performed 2-photon recordings of singing birds. We've got these guys to sing on the microscope, which is no small feat. They'd rather not do that. They'll do that in their native Australia. We put in GCamp just with the virus, and so these cells are all expressing a calcium indicator. You can see that they make little blips when those cells are active. It's actually a really great place to do calcium imaging because all the cells here are very near the surface, and most of them, especially these RA projecting cells, are only active once. So you have a baseline of 0 firing, and then this big burst that's going at 600 hertz and then back to 0. can look across trials and really pinpoint with a remarkable amount of temporal precision when each one of these neurons is going, so we can test hypotheses about how networks are organized. And in fact, this is the data that we have. It's in one plane for one bird. We've done this across 8 birds. I'm not going to show you all the data. All the data are consistent with this bird here, so I thought it'd be easier to just give you what one looks like. So let me explain what we did. So each dot here represents the position of an individual neuron, and I've colored that neuron based on when it's active during the song. So each one of these blobs is called the song syllable, and so this blob, which is purple, all the purple neurons are active during that syllable, and so on. There are a few basal ganglia projecting cells also in this data set. We decided to include them as well. Some of those can burst twice, so you'll see some of these guys are split horizontally like that, and both of their time points are represented. But what's clear is there is no clustering at a syllabic level. We don't see, for example, everybody from syllable A living in this area, syllable B living here, and so on. I really don't know if that's the right level of analysis to consider here. What we really want to do is look at sequences that are basically at the level within a syllable, so we looked at that too, simply by quantifying where within a syllable these guys are active, the different color scale. This is at the beginning of any individual syllable, and this is at the end. And we didn't see any kind of propagation activity or any spatial clustering there as well at this subsyllabic level. So when we come back to these models, what we see is something that really fits possibility 3. We're getting rigid sub-millisecond timing precision at the level of synaptic activity and at the level of bursts, but when we look at representations in the network, we don't see any hint of that at all. So precise behavior in spite of sloppy representations. Okay, so let's dig a bit deeper, right? Cells are not little tiny blobs. We can look at little tiny blobs and understand when cells are active, but cells are complex things. And so what we decided to do is go into our field here, find a cell, and fill that cell up with dye. And so we could look at the structure of that cell, look at its axons, and look at its dendrites. It's a little hard to see here, but this is the outline of HPC in this dashed line. The dendrites are in white, and that red structure here are all the axons from a single neuron. So we can look at these axons, and maybe we can get some hint into this very Swiss watch kind of behavior of these cells by looking at the axons. Maybe there's some specialization that exists out there. And nope. So we don't see anything like that. What we see is this sloppy hardware underlying this millisecond machine. And so what do I mean by sloppy hardware? We have heterogeneous axonal projection patterns of these HPC neurons. Some are small, some are big, some are blowing up and to the left, some are kind of sending projections everywhere. These polar plots are meant to tell you where the weight of each axon is being sent. And they almost look like hands of a clock, all telling different times. So we don't really see any kind of clear, rigid, military-like structure that exists at the level of these neurons. So we decided to look a little bit closer in at these wires. And so I think connectomics is a hot button issue, but I think I would argue, at least in the next few slides, this is where we first start to see structure that can start to explain the phenomena that we see at the level of individual cells. So, of course, this is a neuron, and I'm showing you all of the axonal length from that one neuron. These are tough experiments. We have to fill them up and then take the entire volume into account and take every inch or micron of these wires. And in this case, we've actually also annotated where synaptic boutons are. So these are the outgoing connections from these cells. So this guy is talking to all of these neurons and doing so at all of these little silver spheres. And what we'd love to do, our kind of dream experiment, is to fill in those silver spheres, and we know who these guys are talking to. Right? But that's hard to do just using a standard dye. So how do we get to that? So this is an idea of kind of blow up of these silver spheres and some wiring, and we'd love to color these in. So to do that, what we've done is worked with Winfried Denk, and so he's a person who made his name in two-photon microscopy and has recently pivoted to this kind of serial block phase approach. This is one tiny part of our HPC volume. That was a cell body. This is another kind of cell body here, one that's labeled blackly. We put in a retrograde tracer to actually label these premotor neurons, so we know where they sit within this volume. And as I mentioned, this is just one tiny part of a larger volume that we've reconstructed to try and understand the wiring patterns of the bird. And what's nice about that is, this is a little hard to see, but trust me that there's an axon here. And those same little silver spheres are shown here with this kind of swelling. And those are the connections that this axon is making. And what we can do in EM, because we're free to do anything we want, is just to leap across the synapse and ask what kind of cell is there waiting for us on the postsynaptic side. So we can now decorate that axon with the target of each one of these mutons. And so, for example, we've reconstructed this guy and the postsynaptic guy, and you see it's kind of a featureless tube, if you like. There are no spines there. And those of us who work with inhibitory interneurones know that these are often sparsely spinous or aspinous. And indeed, this is an interneuron connection. So this premotor cell is talking to that interneuron. And in fact, if we consider the dendritic structure of all three of the major players that exist within this area, here's the representative interneuron, and the spines here with these little blue dots are really very rare. This is one of the cells that Dan was talking about, these basal ganglia projecting cells. There's a spine about every micron. It looks kind of like a big cluster of grapes. They're very easy to identify as well. And then these guys, the porridge that Goldilocks chose, this is just a spine every 5 microns or so. And in fact, you can perfectly classify these guys based on simply spine count. So now we can go in and decorate each axon and know who it's talking to and where. And it looks something like this. And so what we found is that inhibitory targets, connections onto inhibitory interneurons, are really prevalent very near the cell body. So if we look at the distance from the soma, the relative number of synapses is almost 1. So that means that almost every synapse is being made onto an interneuron nearby. So we color this blue for interneurons because we're near the soma, and that's the kind of connection that exists. But if we start to go away from the cell body and look at these connections out here, what we find is, and this is a little hard to read, but trust me, that there are a lot of connections onto other premotor cells. Right? So the kind of the connections that are made intrinsically here that could help to propagate a pattern of sequences at a network level really exists far away from the cell body out here. So we can decorate it with this kind of red blob. So the idea is we finally have found some order. It's orderly synaptic patterning of these premotor targets. And the structure is in the wiring logic. Right? So it's not in where these cells are. If we look at two-photon microscopy, there isn't really a clear map. It's not where the actual axons are going. They seem to be blowing in the wind. But what is really a clear piece of structure that we're starting to pull out here is that these guys are talking to interneurons near their cell body, and they're talking to other premotor cells as they move away from their cell body. This has also been shown recently in a paper that came out about 6 months after ours in the medial entorhinal cortex, grid cells. And they find exactly the same connectivity scheme there, where if they look near the cell body, those cells, these kind of grid cells, are talking primarily to inhibitory targets, and as they move away, other excitatory neurons. We also made an observation along the way, because we have the data, that local axons are actually really thin and unmyelinated. Right, so this guy is it's a premotor cell. It does 2 things. It sends a descending axon to RA to help kind of create the song. But it also sends these local axons that I've just gotten done talking about for the last 10 minutes. And it's a little hard to see this, still kind of bright in here, but I'll say that local axons are this distribution. They're really, really small. They're invariably unmyelinated. And these descending axons are much larger and often myelinated as well. So what we'd like to do is get some idea for, we started immediately thinking about delays. And so there are a lot of stories, for example, in the bar now about the importance of delay lines for patterning and for neural function. And we thought that these could represent an interesting source of delays. So what we could do, we couldn't go in and measure how long it takes for a spike to go from the cell body to any other place within this axon, because the axons are super small, really hard to patch. Maybe with a voltage indicator we can get there, but it was tough. But these guys that go down to RA, and I hope you can see in this schematic here, we could actually stimulate them and record an antidromic spike, a spike that's going the wrong direction up here. Look at the conduction velocity and use that to help estimate, given what we know about these diameters. Right? So this is a local axon, very small, this is very big, and use that to estimate the delays involved in talking to different parts of this axon. And this is what I'm talking about. So here's a cell. These are all the local axon collaterals of that cell. This is how far they are from the cell body in the color code. So these are very near the cell body in yellow, and then as we come out here, this one is over 1 millimeter away, and we get several millimeters in some cases. And we can use that kind of antidromic trick to now estimate how long it takes to reach, for a spike to reach any part of this axonal field. Okay? And you can see out here, it's more like 7 milliseconds, and we have cases where it's as many as 20 milliseconds. So there's a huge range of delays. And I've told you already that the temporal precision of the network, at least at the level of firing, is less than 1 millisecond. But we have a huge range of conduction delays. So how does this all work? Something's got to give. So the interim conclusions here are that sequence-generating circuitry can seem unexpectedly sloppy. It exists without any structured network representation. It relies upon distal connection with a range of often very slow conduction. And the next step is to try and create a model. So without Adrian here, I felt that I should pick up the mantle and try and put a model here to understand what we're doing and try and solve this apparent paradox of real cell precision at the level of individual cells, but networks that seem to be kind of sloppily constructed. So let's go into our model. I've given you all of the components. The cellular organization is something that we'll consider here. The spatial distribution of synapses, and this kind of temporal distribution too here. And so what we've done is we've created something with 20,000 premotor neurons as well as some interneurons to keep it from running off the rails. We don't want to have a giant model of epilepsy. And so we have random spatiotemporal structure that's also inspired by what we saw in the experiment. We know where these guys actually talk to other premotor guys, so this is coming from the connectomics work. It's on this kind of more distal side, and that's built in here in the kind of tendencies of synaptic connections. All of that is taken into account. And finally, a range of conduction delays. And our strategy is to compare the modeled data with this recorded population activity and trying to understand how this network works. So our first attempt is a random network. I'm on the same burrow as Larry Abbott, so it's the first thing we should try. And Kanika is a friend, so she came down and gave a lab meeting on random networks. We were inspired enough to give it a shot with all of our constraints. The idea here is that these synapses are formed not independently from each other, but they're in this network of 20,000 neurons. We can start the party with 200 neurons and just see how things propagate through this network. And I should say that each neuron makes 170 synaptic connections, and that's quantified from our EM data. And this is what it looks like. It's a little hard to see because not much happened. I'll blow up this area down here, and what you can see is that the sequence itself fails. We try and prime the pump, getting these 200 neurons to start, and what we see is this very quickly dies off. It really only propagates through less than 1% of the network. So we decided to put our thumb on the scale a little bit and try and help it along, give it a stimulus package, and what we find is by increasing the excitatory synaptic strength of these neurons, we could finally get some propagation, but it wasn't the kind of propagation we see in this network during singing. We see a lot of extra bursts that are happening out here, and we also see that things kind of die out with time. So we don't see, for example, a series of bursts that operates in a diagonal here, but something that really is hard to maintain. So we then hypothesize that this kind of network propagation requires convergence. And cells need to help each other out in a noisy world. And so the model that's been proposed and in the Songbird literature, but obviously before that by Moshe Avalis and others, is the idea of a synfire chain. And so that's one way of trying to propagate a signal through otherwise noisy networks. And the idea there is that we start with 200 neurons per layer. Neurons can exclusively form connections with just subsequent layers here. We've pruned it a bit to try and match some of the experimental constraints that we have in talked about in the previous slide. And when we did that, so here's the intuition, right? Of course, that we have these kind of relatively synchronous groups of cells, each time step here. And so this blue cell will fire, and when it does, this is almost impossible to see, but there's a blue cell here, and there's a PSP. When the yellow cell fires, there's another PSP, but because these are all firing in a coordinated way, that PSP becomes, and trust me, a burst. Okay. So we tried this model, and what we find is something like this. So there's groups of cells that are active, these kind of time steps, and they're active in these blocks like this, right? So those bursts are happening all at around the same time. We did incorporate delays here. Surprisingly, this propagated really well, even in the face of those delays. And if we look at the onsets of when these groups of cells, you can see it almost forms this sawtooth pattern, right? There's these discretized steps, ticks in a clock, if you like. And those are at very specific points in the network, and the time between those is maybe 7 to 10 milliseconds. So the prediction here is that the sequence should be composed of discrete steps. And so that's a testable prediction. We went into the data. We have 2-photon imaging in 9 birds. We also have a data set from Michael Fink, where he's done extracellular recordings in 5 birds. And we can ask, okay, so when are these events occurring? Do we see any evidence for these kind of discretized ticks in the clock? And so these are the bursting events. They're happening kind of all the time. I didn't show the events that are occurring between the syllables, but there are also bursts there as well. And so what we want to do is compare the data that we have with predictions from our model. These are the syllable boundaries. So what I've done is taken a look at every single one of the syllables, both in the electrophysiology data set as well as the imaging data set. This is the actual recorded data when the bursts are occurring. Those are the white tick marks. And the synfire chain model that we've created has very specific predictions, and those predictions are shown here. You can also see it here, but it's a little bit more sparse. It's harder to see. So this, because there's more data, you can see these streaks of spikes that come down like that. And those streaks of spikes are caused because of these kind of simultaneously active groups shown like this. And so if we look at the predictions made by this in frequency space, right, both in imaging and in electrophysiology, we'd expect to see a bump in the data at around 120 or 140 hertz that represent the action of simultaneously active groups. And if we overlay the data shown here in this red line, what we see is a total lack of this resonance frequency, if you like. So this synfire chain network is inconsistent with what we actually see in the data. We don't see any evidence for ticks in the clock. We see that activity is pretty flat across all of these frequency ranges. Okay, so let's try it one last time. And so we found a paper from Eugene Mizikevich, and the idea of a polychronous network. And I think it was a really interesting paper. The idea there is that if you have a postsynaptic cell, for example, represented by this vertical line, it can be receiving information by cells that actually start it at very different times. So imagine this cell is firing at time 0, but it takes a bit longer to actually reach this postsynaptic cell because of some delay. As I mentioned, we have these kind of slow, unmyelinated axons that could be a perfect source of exactly this delay that Eugene speculated many years ago. The cell is active at time 4 milliseconds, and this guy is active at 8, and yet they all converge at the same time and cause this cell to fire. And so the idea in building this model, we have all the same experimental constraints that I showed you before, but now we're lacking these discrete coactive time steps that were seen in the synfire chain model. So this requires convergence at the level of individual neurons. The idea is that just like in Eugene's case here, if there's a blue cell that's active here, causing a postsynaptic response, another cell that's active here, but with a delay, they both can reach at the same time and create a burst. So we tried this model, and indeed what we find is something that produces a series of spikes that lacks these kinds of steps that have been seen in our synfire chain model. See them blown up here. And the onset timing lacks that kind of sawtooth pattern that plagued our model, was inconsistent with our data set. And so the idea here is that this polychronization model creates a continuous burst throughout the sequence. And so we now compare the data that we've collected from imaging and from electrophysiology to the predictions made by our polychronous model. These match in a kind of qualitative sense very nicely. And we've also done hypothesis testing on this, and what we find is the polychronous prediction is just as flat and lacking the kind of frequency resonance that the synfire chain had, and matches nicely with our data. And so one question is, how can this network help the bird? Right, so as Dan mentioned, the concept of vocal gestures, this is something where different parts of the song can actually be discretized because these are pressure and tension transitions. And we can look at the actual timing of the vocal gestures. Gabo Midland took 8 of our birds and pointed out where the vocal gestures were in these 8 birds, and we were able to look at the relative timing of these vocal gestures to ask, is there any kind of periodicity to those gestures as well? Right, that would be indicative, for example, of a synfire chain model. The prediction of the polychronous model is that one can actually put a vocal gesture at any point in the song, whereas the prediction from this kind of synfire idea is that they should really be relegated just to specific time points that are set by this resonance frequency of the model. And what we find is that things are really flat, that a bird is able to sing what he wants, whenever he wants, and is not restricted into where he can put his vocal gesture and actually put that at any moment in the song. So that's something that I think is a feature of the polychronous network that is not a feature of the synfire network. Okay, so in wrapping this up, sequence generating networks can exhibit circuit dynamics that are well explained using this polychronous feed-forward network architecture. I think these guys don't cut it when we try to take all of our experimental constraints and place them into these models. This one really shines. These guys don't match the biology. And the network lacks this kind of time step or clock that would be present in a synfire network. And this reminds us of asynchronous computing. This is computing without an explicit clock. And the advantages of that is it allows for faster and more flexible networks, well suited for applications where messages are generated at different intervals with a range of delays. So I think these delays that we see are actually a feature and one that can allow this network to be built. And what we're trying to model now is how this can actually help the bird when he's building this network through development. And that's something that's ongoing work, and I'm happy to talk about that offline. These are the people who did the work. I worked on the model with Deja Shin and Eugene Tubikov at Penn State, and collected data with a lot of other people. And thank you very much.
That was fascinating. In other words, the sloppiness leads to flexibility. That kind of thing. That's
Yeah.
kind of fun. Questions?
Someone over there or Barbara?
So I have a question about simplicity. So at some level, all your modeling approaches are based on the notion that in some space things are simple. But if you remember that great, the world is learning over time, and it's learning a song. And a system that fine-tunes all its synapses will learn to be good at timekeeping, however timing mechanism it uses. So that means that the actual mechanism could be very different to what you have. It's like replicating certain aspects. How would you justify that you believe it's simplistic in a way that us humans could understand? No, because the whole history of how we get there.
40,000 dimensional space. So I would say this isn't an asymptotic case where the bird is an adult and can learn itself. How this works during learning where things are more flexible, I'd love to know. But I want to know what assumptions we made here are simplistic. I would argue we've dug extremely deeply into axonal structure, conduction, connectomics. So I think we're...
Look, it's like beautiful analysis, don't get me wrong here. It's just that these three sets of models that you set up, they're at some level, they make very simplifying assumptions, and it's violated in the data. Your model predicts that each cell will produce roughly the same sequence within a burst. And
I think that what I can do, and maybe this is also simplistic, but I think the flavors that we're trying to go for, random is a flavor, and we can just put that aside for now. But we can say, how can we get convergence? So one idea is that convergence is something you get for free, because something like a synfire network is going to have convergence because things are synchronously active at each time step. So necessarily, at a presynaptic level, you have groups of cells that will fire together and they converge for free. Whereas the idea from Izikevich, right, this polychronous idea, is that cells can start whenever they start, but when they come in is the important piece, right? So in some sense, it's a postsynaptic view of convergence. So those are the kind of flavors that we're going for. Is this what can allow a network to work? Is it coming from presynaptic setting things in the architecture at that level, or we just kind of let it go ad lib and let things kind of come in at the level of the postsynaptic neuron? Does that make sense? So those are the two flavors. I'm happy to discuss more later. Yeah.
So, do you think that when the bird sings, the interneurons produce lots of spikes? Lots and lots of spikes also. So do you think that those interneurons are not contributing to the timing in any way? Or is it something you're going to try to explore more?
We're going there next. I think this is kind of zeroth order.
I don't know.
Yeah, so the interneurons are there. They represent 10% of the network. They're firing, and instead of this very cute delta function where things just make this high frequency burst and then the rest of the time is silent, interneurons are really kind of running all the time. And
But also...
yeah, they're varying their rates as well. And we're absolutely going there. We're very interested in how the interneurons can kind of work with this structure in order to clean up errors, for example. And I think that the interneurons are a very big piece of the puzzle. And I think trying to understand this backbone gives us a framework to now layer on interneurons, which are an extra level of complexity. But I think it's in addition to this, as opposed to something that's fundamentally different. I think interneurons are doing a ton, for sure. But I think we needed to understand this first.
That's fine, but why do you think that it's adding on a layer as opposed to potentially changing the structure? I mean, you've shown that.
So,
So I'd love to hear more about that.
yeah, so if I think interneurons are helpful for understanding this. But I think there are a lot of connections, and some of them are nullified. Right? And I think, why is it that these all seem quite big? I didn't put a scale right here. They're 10 millivolts. This guy makes a big burst of spikes that comes in at several hundreds of hertz. These guys are totally ignored. And I think what we have is a network where there's convergence, probably through a polychronous style wiring strategy. And then on top of that, interneurons, which are firing a lot, are keeping these guys from creating bursts. So they're actually helping to sparse the network. I think they're doing a lot, and I think they're extremely important. But there's also a diversity of interneurons. I mean, the mouse people are way ahead of us on this one. So the idea of dendrite targeting interneurons versus soma targeting interneurons, interneurons that target other interneurons. There are a lot of different kind of types of logic that interneurons are capable of. They're extremely diverse, even in the bird, and we're exploiting that diversity now, and we see several categories of cells that could be doing several interesting things with this, and we'd love to play with that. Yeah?
What determines the width of this polychronous line?
Repeat the question.
What determines the width of the polychronous?
Well, the spread.
Oh, right, okay, thank you.
Spread.
So there is one thing that I didn't talk about here, but we talked about pretty extensively in a 2010 paper, and that is the presence of a calcium spike. So one reason why this guy actually makes a big all or none burst of spikes is because these dendrites are actually capable of generating local kind of short calcium plateau potentials. And so I think that you get enough synaptic input onto one dendritic branch, it fires, right? And so I think the width of it is kind of, in some sense, a property intrinsic to the dendrites. It's an integrated burst, if you like. And so that I think is, and we build that into the model as well. A two-component, each cell is a kind of two components. There's a dendrite component and a soma component. We give L-type calcium conductance to the dendrite, and we have physical reasons for doing it. So I think that sets the actual width, that kind of 4 or 5 spikes in the burst. The problem is, without this calcium spike, things can really blow up or die down. So to actually propagate a burst is a tough thing to do. And we showed this in a previous paper. Does that answer your question?
Maybe, but you have this graph where you have like a lot, in a synaptic chip, a series of blocks, and in a polychronous state it was a line. So yeah, right there, slide 41. Yeah, so this is for the synaptic here, but for the polychronous,
Right.
heterogeneous.
Right, and what you'll see, these are about 4 spikes per burst, give or take, right? That all or none bursting is a feature of the calcium spike. And we can modulate that pharmacologically to keep L-type calcium channels open longer and burst get bigger. We can knock them down and burst get shorter. But so that's what sets the actual kind of wavefront of these things. Yeah.
So one really interesting thing about the polychronous thing is that synapses kind of only go forward in time. Like every cell will excite cells that are forward. So if you would like inject, say, rabies into the last cell, could you see the waves over days kind of go back through the entire sequence?
Right.
So super cool.
No, I totally agree. I'd love to do that. I think it's probably more complicated. I don't know what actually sets the refractory period, why these guys are only bursting once and not more, but it could have something to do with the interneurons. You can imagine a stray wire that comes over and wants to put something that's before this in time, right? If they kind of, then the interneurons think, that's not right, we're going to turn this off, right, in a sense. But yeah, it would be a lot of fun to see the structure, higher order structure of these guys holding hands. I think of, yeah. Yeah.
Mike, so the thing I'm kind of, this is Conrad's first question I'm going to try to pick up on. So you started the talk by, looking for structure in effect, right? And you could view it as, well, humans would like to see some structure, and then you go down and go, actually, it isn't quite so clean. But then I like the approach that you said, well, let's put in kind of minimal ingredients that we need to get this to happen. That's sort of how I read your modeling thing. Start with the random. But I still don't have a sense of what you think, the polychronous model, what ingredients need to be essentially built in versus what are learned with a reinforcement signal. And is it, I think that's kind of where Conrad's first question is pushing.
Okay, so you're bringing in learning and reinforcement, which is, I think, those are a lot of themes that exist in Songbird. And I think what is pretty interesting is that Michael Fee has now filmed neurons in birds that haven't been exposed to a tutor. So kind of early activity to ask what's the tendency of an early network before the animal actually hears anything. And he starts to see this kind of sequence that is already there. It's almost like pre-play or something like that, right? And then layered on top of that, when the bird starts to sing, the bird takes advantage of this pre-existing sequence. So I don't know how much reinforcement is actually involved in setting up this timer, right? It could well be that there's already a tendency. It's just in there itching to form a sequence, and then when the bird sings, it finally goes full bloom, in a sense, right? But I think how to develop this is something that we'd love to know, and we're playing with it. But I think not having to set up this scaffold structure like a synfire chain really frees up the network to develop itself much more robustly. But we can talk about that offline.
More thoughts on that?
The polychronous model, does it have, could you talk about its sort of capacity? Could it multiplex timing multiple signals at once? How does the new input disrupt ongoing activity?
So Dan hit the nail on the head earlier. I mean, we're working on, really a zero-noter bird at this point. The bird is very, very simple. It sings one thing over and over its whole life. It's bad at it at first, and he gets really good at it, and then keeps that way for 10 years and then dies. And so that's we would love to know, even other birds, related species, which is a Bengalese finch, actually mixes it up a little bit. There's a little jazz there, where if you know this note, it could be that note or that note following. It's not always this deterministic thing. So we would love to know how cells are reused. Obviously, the mammalian motor cortex is a very multiplexed area where cells are kind of brought in to do a lot of different things, right? So this is a very specialized case. And what we would love to do over the next few years is to try and get less and less specialized, where we try and push the cells to do more and more and ask exactly those kinds of questions. So if a cell has to wear different hats, how does it do that? How things get routed through like that? I think it's a great way of thinking about it. Ben?
So it remains unclear to this day to what degree are the basal ganglia projecting neurons in HPC important for singing. There's, as you well know, but maybe the audience doesn't, there's even a paper out there that claims they're not important at all.
Yeah, that's an extreme view.
And I'm curious, but you have a lot of beautiful data that, given the totality of it, what's your current view about that? What are the experimental
Right, so Dan brings up the point of these basal ganglia neurons. Constance Scharf, 18 years ago, poisoned them, killed that population off. Of course, they're very important for acquiring the song, but in the adult, the bird appeared to sing just fine. The method is a little shaky, and it hasn't been revisited in a careful way. I don't know. I would love to know. And I'm not putting my head on any chopping blocks right now.
No, I'd love to know.
Yeah. I'd love to say they're not involved at all because they're not in our model, But I don't want to go that far. Interesting question.
But if we think about, let's say in sensory systems, we have primary sensory systems, and then we have non-primary, going up through the thalamus and to various parts of cortex and V sub X and A sub X, et cetera, et cetera. So if we think about descending systems in complement mode, there might be primary systems, which may well be HPC2RA, And there may be non-primary systems which carry either sort of higher order information that's not necessary for immediate performance, obviously the acquisition, but maybe they kick in when the delayed feedback comes on or something like that. In other words, they're there to break it. After all, when you do force changes through interference feedback, which is one of the systems where you can basically negatively reinforce certain song patterns in order to drive song development, then you see temporary reorganization and then later reorganization in the pathway that involves the begin. So,
Right, no, you're exactly right. I think it's well
does that make any
positioned.
sense? Yeah.
No, it's well positioned to do something like that. I mean, slow changes could very well be mediated by these predictor guys.
I thought the battery was still working.
Yes.
Be loud.
So just simply to add, I mean, it's, I think, important for everyone to appreciate that the song of this very precisely singing bird is constantly being modified every day. The only way the animal maintains its precision is through constant modification. And there's an argument
And
that
sleep.
there's an argument that sleep modification during sleep is an important part of that process. So this is not, it may be ballistic when the animal is doing great and singing one song and not perturbed, but there's constant learning taking place, just like any great musician or athlete or anything like that. There's learning to maintain that seemingly ballistic behavior. It's important to appreciate that.
There's modification and anti-modification such that if you take snapshots at the same time of day over time, you see what appears to be stability, but actually it's dynamic.
Yes,
Right.
yes.
Randy?
You started the talk with this apparent contradiction between individual neurons having very precise spiking patterns given their input, but then seemingly random behavior of the networks. Why? Why do we bother having thousands of neurons doing seemingly the same thing if you maybe could have just a bunch of them doing the same thing? What do you think is the purpose of the complex network?
I think that I'm not a sensory physiologist. There are some extremely preeminent sensory physiologists here that could probably answer this. But I think the problem is, I put the study up about adding a neuron, adding a spike and getting 28 extra spikes, and we're in chaos here, people. I put that up there as a kind of false note of caution. I think as people start to do the right kinds of experiments and consider the right kinds of sources of variance, what they find is alarming amounts of structure. So, I mean, David McCormick has some beautiful work where he started to see a lot of variance in his data in auditory cortex. He was patching onto cells and giving some auditory stimulus. But then his postdoc noticed that the pupil diameter actually changed quite a lot, the level of arousal. And once they controlled for that, it really explained an enormous amount of variance that they started to see. So you can imagine hunger state, arousal state. Michael Brecht has beautiful data showing that estrus cycle state can change the baseline firing of interneurons by a factor of 4. Factor of 4, fast spiking cells. That's amazing. So there's an enormous amount of variability that once we start to appreciate and correct for, and that's why I like the cat experiment, because there we have full control. Freniak has full control over what that cat is seeing, and can play this back in exactly the same way. And what we find is the appearance of structure. Things are not noisy. I think we've been very quick to say, I don't understand it, therefore it's noisy. In the same way, I don't understand this medical condition, therefore it must be possession, and not, for example, mental illness, right? And so I think that noise and people who kind of perseverate about how noisy the brain is, it's a bit religious.
Yeah, there's a direct follow-on to that, because unless I misunderstood you, were arguing that it's just noise in those axonal distribution patterns. And I was thinking just what you were saying. Well, we keep discovering that what we think is noise is actually very intricate structure. So
Exactly. So I think that when I first saw those axon patterns, I thought, oh man, this is a much more difficult problem than we thought. But then when we got more kind of micro about it and saw that they actually link up in a very clever way, right? So that they like to make these connections out here where delays are a factor. They could actually use this delay. It's no longer a bug, it's a feature.
no, I understand that, but you left me at least with the impression that, as you said, the actual pattern of distribution of those axons was blown in the wind.
Yeah, yeah.
It was essentially random.
Yeah.
How strongly committed are you to that?
I think it's pretty, I mean, we have 22 of them. We've looked at this and there's no regularity to that. And we thought we saw something along the lines of what Dan saw too, where we looked at two filled cells in one bird to ask if, for example, there was some kind of wiring motif that could be kind of conserved within one brain. And it's a hard question. That's not powerful enough, I think, to answer that question. But it would have been a cool thing to see. It didn't immediately leap off the page. But I think Dan's doing the right kind of experiment looking at the kind of biophysical level. Yeah.
But isn't there an issue here, like the issue with spike patterning and what's the neural code, that the more efficient the code is, the more nearly it will look random? Isn't there?
You have to ask Conrad about that.
Well, no, but it's a profound truth, right? That is that it doesn't. Of course, it leaves us not knowing. Well, is this actually random, or is this?
I think everything's complicated. When I started graduate school, people were looking at the medial entorhinal cortex because you patch onto these cells in slices and you hold them just below their threshold. There's some kind of subthreshold gamma activity, and they're staring at gamma like it was Jesus. And then finally, the Mosers put in a wire and see these guys running around, and now there's this whole crystalline map of space that wouldn't Nobel Prize still don't understand. But the point is that, I think finding the cell in its element, I mean, and by that I mean behavior, I mean, is really to understand what the constraints are. I mean,
Well, that seems like a profound observation and a good time for lunch. Thanks a lot.